those of you unfamiliar with the Amphibian-Killing fungus, it is the newly emerged pathogen of frogs and salamanders, caused by the aquatic fungus Chytrid dendropatitis. In our study system in Brazil, we see two deeply divergent lineages of the Amphibian-Killing. A putative Brazilian endemic that's deeply divergent from the lineage we know to be responsible for outbreaks of disease around the world, or global pandemic lineage, which I'll abbreviate GPL. In Brazil, our prior population genetic work has suggested that these two lineages co-occur there due to secondary contact, most likely through the introduction of the global pandemic lineage through human activity. So we wanted to reconstruct the population history of this coexistence zone and to understand the consequences of the secondary contact. And we did that by whole genome resequencing 51 strains that we isolated from infected amphibians in this area. This diagram is showing genetic relationships between those 51 strains inferred from distance across 90-some thousand SNPs, and the associated structure analysis for these isolates on the right. You can see pretty quickly the deep divergence between the global pandemic lineage here in red and the endemic Brazilian lineage in blue. But these are not the clones you're looking for. I want to draw your attention to 3 hybrid strains that we recovered from the Atlantic Forest region of Brazil. I should mention that this is the only known instance of interlineage hybridization in this species. The structure analysis suggests to us that we have 2 F1 hybrids that appear to be independent of each other, and 1 F2 backcross to a Brazilian parent. With our genome data, we're able to look deeper at the patterns of inheritance in these 3 hybrid isolates. And for this first analysis I'm going to show you, we identified SNPs that were fixed in the global pandemic parental population, SNPs that were fixed in the Brazilian population, and looked across the hybrid genomes to identify positions that were homozygous for one or the other of those fixed SNPs. You see, qualitatively here, our 2 F1s show a lot more red homozygotes than blue homozygotes. And this is significant because we actually know very little about the process by which eukaryotic pathogens diverge and locally adapt in allopatry. And we believe that at least some of those loci I showed you that were unequally inherited from one parent or another were inherited in that pattern due to incompatibilities that arose through this divergence process. So we hypothesized that at least some of those loci are inherited as such because of either incompatibility or selective instability, and could provide clues to the divergence, the genetic underpinnings of the divergence between these 2 lineages. I'd love to talk a lot more about the system if you're interested. I think I may have time for 1 or 2 questions, but if you're not able to ask a question in the session, please catch me after sometime throughout the meeting. Thank you guys for your attention.